we can kick off the remaining 2 sessions of the symposium, our panel discussion up next, followed by the Meet the Speakers breakout sessions. All right, well, we're waiting for Nathan to turn his camera back on. I will quickly introduce one of our panelists that has joined us specifically for the panel discussion, also the Meet the Speaker afterwards. Andy Yeung, who is currently the Chief Technology Officer at Asher Biotherapeutics, a biotech startup developing targeted cytokines for cancer immunotherapy. Previously, Andy spent 10 years at Pfizer, and where he was most recently the director of biotherapeutic design and discovery, and led a number of engineering efforts, including the development of CD3 bispecifics, CAR T platforms, and immuno-oncology programs. Prior to Pfizer, Andy did his post-doctoral research at Genentech, actually, and received his Ph.D. in chemical engineering from MIT, also in the lab of Dane Wittrup. So, Andy, thank you for joining us for the panel discussion. All right, so for the, yeah, of course. So for the panel, just for everyone in the audience, feel free, we have prepared some questions for the panelists, but feel free to use the Q&A as you'd like to put questions in, and we'll try to get to as many of the audience questions as possible for our panel. And then again, for those questions that we can't get to and the speaker can't answer during the Q&A, feel free to join the session, the Meet the Speaker session afterwards to get some of your questions potentially answered. All right, so I will kick it off with the first question. And just for this question, I think we'll go quickly through each panelist, and then from then on out, just in the interest of time, we'll maybe take 1 or 2 panelists that answer. But for the first question, kind of just big picture, if you had to pick a cytokine today to deliver therapeutically for cancer, what would be your favorite cytokine and why? And just in the interest of time, we can limit the responses to just a couple sentences. Jamie's smiling, so why don't we start with Jamie?
Yeah, hard to choose just one, right? There's so many in combination versions and things like that. I would say, I mean, I'm definitely biased, and no pun intended there, but I'm a big fan of IL-2. I do like that having the 3 different receptor chains really adds a lot of color to that molecule, and allows for more selective and nuanced targeting. I think compared to some of the other cytokines that have only 2 chains, and also not having to deal with like hexamers or dimer sort of situations, I think just makes it easier a little bit to manipulate. But again, I think there's many shots on goal in many approaches, but I do think IL-2 definitely has a lot of potential.
Great, thanks, Jamie. Let's go over to Andy.
Yeah, I would just say I would agree. IL-2 would be my choice too, both on functionally and also clinical experience. As you might all know, IL-2 is one of the 2 clinical approved drugs. And I think the more exciting data recently is from several studies combining high-dose IL-2 in PD-1 failure patients, where they see similar type of response, 20% ORR, 10% CR in these PD-1 failure patients, because these will be the patients that, like the new line of therapy will be aiming to treat. So I think those are, exciting times. So it's clinically validated. I think the panelists today have probably alluded to, I think with cytokine therapy, we just need to kind of segregate or decouple the efficacy from the toxicity. So with IL-2, that's certainly the case, where if you can make it safer, retaining its efficacy, I think clinically is proven to work.
Great, thanks, Andy. I'll move it over to Jeff. See if we
And
can.
not as a surprise, since I chose to talk about it, but I really like a cytokine that can generate other cytokines, or a cytokine that can generate other chemokines and have kind of a snowball effect. So, if one, as that IL-12 is a good example of that. As I said, it's not a surprise that I would say that. But I do like it because of the snowball effect. And as Andy said, you just have to master the toxicity of the molecule in order to be able to move that forward. But if you can master that toxicity by targeting approaches, I think IL-12 is very interesting based on that.
Great. Thanks, Jeff. And Michelle.
It's hard to choose between IL-2 and IL-12, but I think I'll go with IL-2. And again, like, we definitely need a way to limit its toxicity, either through maybe some kind of masking or an approach that you, Jonathan, developed with the Ortho-2 system in being able to specifically deliver it to CAR T cells.
Great, thanks, Michelle. And Nathan's back just in time. Did you hear the question, Nathan?
I think I know the question. Is this what's your favorite cytokine and why?
Uh-huh.
Yeah, sorry about that. I had to run into the lab to address something quick. But now I'm back. Yeah, I think to me, actually, there is no easy answer, but the answer depends a little bit on the context, right? I mean, I think if we're talking about just a straight ahead cytokine that's meant to have single agent activity, perhaps, and elicit tumor control, then I think, yeah, something like an IL-12 is a great idea if you can tame the tox, right? But in another setting, though, if you're trying to use cellular therapy, one of the chief limitations there is keeping those cells alive long term so that they can actually maintain tumor control, right? And in that case, maybe pushing them hard into sort of a potent effector state is actually not a good idea, because that is going to make it more likely that they're going to die, they're going to become exhausted, or something like that. So, maybe taking the opposite approach and something like an IL-10 actually could be a good idea for them, just to maintain their viability long term, even if it seems a bit counterintuitive. So I'm going to refuse to answer directly.
No, I think that's great, and I also think you brought up a great point that we'll discuss a little bit further. I do want to take one question from the audience a bit later about combination, right, of cytokines with other immune agents, and maybe there's not a good one size fits all, right? Maybe one cytokine is better combined with a cell therapy approach and another with a vaccine approach. But first, since IL-2 has drawn a lot of attention, both at this panel and also recently in the news, well, why don't we'll take a question from the audience, from Yeung about what the panel thinks about the recent non-alpha IL-2 clinical failures with both the Nectar molecule, the pegylated IL-2, and I guess they're both pegylated, and the BMS molecule. I guess the question is going after a non-alpha the right direction? Or do you think these clinical results are reflective of IL-2 in general as a whole?
I think it's pretty.
Nobody wants that one.
I'm gonna jump in because there's silence. So I think to stimulate conversation. So one thing that we could say about Nectar is that the pegylation domain would fall off, and at the end of the day, it was IL-2, right? I think it's a little harder to say that about the Sanofi molecule, where you've got the stable pegylation, and you should be holding that effector stimulation pretty well and avoiding at least the preferential Treg. So that one has put me more in a negative mode towards this, just as I expected better results with that one than the Nectar, and I don't know that we're seeing it. So I'd love to get a broader discussion of that with the panel. Is do we believe that these other non-alphas that are lined up in phase 1 or phase 2, are they going to be better? Are they different enough to overcome that efficacy challenge?
Yeah, or building on that, do you think other approaches are needed to really unlock the potential?
Is it both? Is it the right combination?
Maybe I could take a step at it, but I think you guys probably won't be very surprised by my answer at all if you heard our talk or our poster before. So maybe I'll start by talking about BEMPEG. I think BEMPEG is, I think what Katherine talked about as well, because once the PEG fall off, that is essentially an IL-2. And if you're looking at the doses and the dose regimen of that molecule, it's almost equivalent to a low dose IL-2, right? So I think early days, people have tried high dose and low dose IL-2. And, people have to stay with high dose because the low dose didn't give that efficacy, right? And ironically, I think a lot of like the low dose IL-2 is being used in autoimmune setting. And also there are company engineering, alpha biased IL-2 that actually biasing Treg. So if you once look closer to, the BEMPEG publication, I think in their supplementary, they actually saw like around like tenfold expansion in Treg in the periphery. So which I think in that angle is very more straightforward to answer that. So for the not alpha, our view is that it biased toward NK cells. So we think that if you look at the profile, a lot of the not alpha molecule all the way from Roche Fab2V molecule, which has since been discontinued, and the CEA2V, the PD profile look very similar with a strong bias against NK cells. So our view is the NK cells is limiting the drug to get to the CD8 T cells and get to the right place, and also potentially NK cell mediated toxicity, which has been shown by our group, and also I think Dane Woodruff's group and also other group that depleting NK cell in the preclinical model seems to alleviate some of the toxicity associated with the IL-2 and also other immunotherapy as well. So I'll just leave it at that.
Great, thanks, Andy. Yeah, so based on that, you really think that biasing it away from NK and of course Tregs, if you want to do cancer immunotherapy, is going to be, will potentially be key to, or could be key to having a better therapeutic window.
I think maybe if we move on from this conversation and broaden it away from IL-2 again and think more broadly about cytokine immunotherapy is we know that systemic administration of cytokines has a lot of challenges, mostly around toxicity and undesired effects. And we've been doing this for 40, 50 years now. What are you see as the new promising new approaches to address those challenges with the use of cytokines as drugs? That may really be able to bridge that divide to actually, providing a real benefit to patients.
Well, I think the examples of targeting that we've seen in the literature, the one that I showed, Dario Neri's approach to immunocytokine targeting, that's been a long slog, but still with some joy there. But I think targeting the tumor microenvironment or targeting the cell surface so as to somehow also then localize in the tumor microenvironment. I think whatever one can do to change the ratio of drug in the tumor to drug in the blood is a good thing. That said, I guess myself, I'm not a big fan of the long circulating cytokines, and some others disagree and are investing money along those lines. But in our hands, in the mouse, even when we take some of the mass cytokines and make them long circulating, they don't necessarily even stay masked forever. So I think that the long circulation is a big challenge, but long tumor residence has to be is a sweet spot.
I agree. I think targeting is one of those approaches that's definitely being tried and trying to get to the tumor microenvironment. Are there others that folks see as being able to help with that systemic toxicity or just to increase efficacy of these therapeutics?
Well, Nathan mentioned cellular therapies. I think that is an approach. Cellular therapies, expressing not only their cytotoxic capacity, but also expressing as other payloads these cytokines that we've been discussing, or these chemokines that we've discussed a little bit. I think that is another approach to targeting by using the cell to target. It's got the complexities of cellular therapy, but I think is potentially very powerful.
Yeah, that's a great point. And also, gene therapy or targeted gene therapy approaches to try to get cells right in the tumor or other places to actually express these molecules themselves, I think also, harbor, potential promise. But yeah, no,
It's a great. No, sorry, I think you finished.
Oh, no, I was just going to say, like, I think in my mind, right, there's multiple levels of targeting. There's the targeting on the level of, kind of the cellular, right, cell type specificity. But then there's also the location, right, targeting. And until we address both of those, I'm not sure how much success we're going to be able to see with the IL-2 therapies or even cytokine therapies in general in the clinic until we can sort of address both of those challenges. But sorry, Jeff, you were saying.
Yeah, no, I was going to say it's a great point you make about gene therapy. And, one of the exciting things about tumor targeted gene therapy with a cytokine is that you don't need to transfect a majority of the tumor cells. Even transfection of a minority of the cells in the tumor microenvironment ought to generate the cytokine milieu that you need to do exactly, what you're talking about. So I think it eliminates or avoids, potential difficult spot in cancer gene therapy if you need to transfect the majority of the cells.
Right. And to your point before, then you don't have to worry as much about the circulation half-life and things like that, because it'll already be where you want it to be, kind of ready to go.
Yeah.
Do we think that approach would still require some sort of tumor tissue homing or retention mechanism though? Because I know, at least from mouse models in our experience, like if you stick something directly into a tumor, it does still get systemic if it's produced at a high enough level, right? It's really quite, with a soluble factor, it's quite hard to really kind of keep it in that TME. So we've kind of thought that, maybe you still need to have at least some aspect of tissue targeting in order to actually retain it in the site of interest.
I think that's a good point. We're in my lab quite interested in that with these matrix binding cytokines. We had done some earlier work with some different matrix binding domains and published on those that are very promiscuous binders to say, like, if it's secreted, then let the glue it down and be retained in that environment to avoid leakage.
I'm curious to know what people on the.
Oh, sorry.
Oh, sorry. Go ahead, Jamie.
No, why not, right? Why not use the better version of the cytokine that has more, sort of targeting? I mean, as long as you're not seeing, any issues with it. But yeah, no, it's an interesting point.
I'm curious what people on the panel think about the idea of pH sensitive masks and things like this on cytokines. This is something that Genentech has kind of batted around for some time, I think, but we've never quite become convinced that this is a path forward. But I know some people have been very against it, some people are for it. I'm curious to know what you guys think. I don't have a clear opinion one way or the other. I'll just say that now.
You have to get it to work first.
That's a fair statement.
I haven't seen it. I think it's kind of the masking thing too, right? Is the mask, the breathing of those masks is not perfect. And once it's off, at least with the current technologies that are out there, it's off and it's essentially unmasked, right? So I think I can kind of see that as the same kind of problem is that you're not getting truly the tumor specificity that you need.
Yeah, and I guess I still also wonder how well protease-based approaches will translate into humans. I do think applying them to cytokines could be a potentially great use case because cytokines are so potent. So maybe even minimal unmasking is useful in that case. Whereas something like, cytomics and their peptide mask CTLA-4, it's a bit different, right? You need large dosing, a lot of unmasking for probably full receptor occupancy. Maybe cytokines, as Jeff mentioned, and many others, right? You just really need to initiate that process, and then you have a huge cascade that occurs. So I guess I don't have a great answer for you. Maybe I'm not supposed to be answering, but if we really believe in the translation between mouse and humans for tumor specific or selective proteases, maybe a masking approach for something very potent like a cytokine could pan out better than something that needs a lot of occupancy within the tumor.
Asking for a cytokine could offer advantages too over some of the developability issues, right? Like if you were able to cover some of those hydrophobic patches, which is where you want to do for a mass, does that help you with your half-life and just manufacturability, even in just the manufacturing of the drug itself?
I know, Jeff, you have obviously direct experience with masking. I mean, you don't have to add anything else if you don't want, but do you have any thoughts on the conditional?
Yeah, not on the topics that have been mentioned about masking, but I think that selecting masks or designing masks with immunogenicity in mind is particularly important too. One could cook up completely unnatural masks, that probably will cause troubles with repeated dosing. So I think masks that are biologic, natural, evolutionary origin are probably beneficial. In terms of pH sensitive, I really prefer proteases, but I think that, Jonathan, you're right. You're just not going to know if they're going to translate well to the human until you do it in the human. There will be, there could be other tumors in larger animals that you could go into, but the real rubber meets the road is going to be in the clinical study. I know one would rather not bear that risk that long. We were somewhat comforted by work with tumor explants, but that's still an explant.
Yeah, no, I think that was quite encouraging from your data set, was showing that you actually enzymes from the human tumor, right? Versus just exogenously added to media.
Yeah.
I'm just looking at the Q&A here. I'm not sure if it's a question or a statement. More of a statement for everyone if they want to read that. Thanks, Francis, for that statement. All right, I think we have about 5 more minutes of the panel left, and we sort of touched on this.
I'm curious, and we've talked about cytokines, but I'm really curious to hear people's thoughts on chemokines. If our objective is to enhance both DC and T cell infiltration of the tumor, what do some of the other folks on the Zoom think about chemokine therapeutics?
Yeah, that's a great question.
I mean, I think the chemokine concept is actually a really interesting one, and I'm glad you showed some of your data on that, Jeff. You guys, using your approaches, because I think that could be a nice way of addressing some of the challenges of like the immune exclusion biology, for example, and things like this, where we may have active T cells, but they just can't get to where they're supposed to be, right? And Genentech has been playing with trying to manipulate TGF-beta signaling, for example, to try to remove some of these stromal barriers and so on to T cell infiltration. But I've wondered as well. We don't have direct data for this, but I've wondered as well if part of the immune exclusion issue is that stromal cells themselves are producing chemokines that are just homing the T cells in the wrong direction, right? They're not getting into the parenchyma; they're going to the stroma instead because they're such potent drivers of these types of factors. So if you could find a way to redirect those T cells to where they're supposed to go using some kind of localized chemoattractant, to me, I think that's a cool idea.
Mm-hmm.
Yeah.
Anybody else?
Yeah, I think it's really neat, an exciting concept, right? Why not recruit, sort of more players. And I think the important thing for me is just, sort of making sure who's getting the chemokines, like sort of which cells in there are able to, access the chemokines most easily. And, are there ways that we can sort of play around with, accessibility of the chemokine to different cell types within the microenvironment?
Yeah. And I also thought it was pretty interesting, and Nathan, correct me if I'm wrong here, but there was also a connection between Jeff's talk and yours, and that Jeff showed CCL4, right? And tumor retained CCL4. And if I recall on your slide where you looked at responders and non-responders in the context of IL-6, CCL4 was one of the genes that came up in the responding population of IL-6 low, if I recall correct.
Yeah, that's right. Yeah, we see a few chemokines. One of the other ones that I think is interesting is XCL1 and XCL2, which are kind of important chemokines for mediating like T cell dendritic cell crosstalk, right? So these are chemokines that get made, by a relatively freshly activated T cell, and then they help draw in sort of dendritic cells to the space that they need to be activated. So I wonder if, and this I think is where, this is all part of the sort of type 1 interferon-like signature that we see in these responsive tumors as well. And I think it maybe represents that these tumors just have the right kind of soil basically to propagate an immune response, right? And the question is, how do you generate that soil? I mean, something that hasn't been talked about today at all is actually using interferons themselves as cytokine therapies. And I think that's a whole big area on its of its own that could be exploited, right? But the same kind of challenges apply, like how do you administer interferons and get them to the right place without causing toxicity? And so it all circles back to the same conundrum. Chemokines, though, I mean, are not themselves acutely toxic, right? So that's an interesting thing, but you do have to get them to the right place. So again, you're sort of coming back to the same problem.
Do you know, and I actually don't know, but while we're on the topic of chemokines, just to kind of wrap it up, what has been the clinical evaluation of chemokines for cancer immunotherapy? I guess therapeutically, have many people tried to harness them in the clinic? And that's an open question to the whole panel.
It may be, but I'm unaware of it, actually.
Yeah, I can't think of any clear examples either.
Yeah. So either
There
have been some like antagonist approaches that I think have been used. But yeah, I can't think of any agonist approaches.
Yeah. All right. We probably have time for one more question. Kat, do you want to do one more question before we wrap up, or a quick answer, and then we can wrap up and do the breakout sessions?
Yeah, I think we talked a lot about the engineered cytokine approaches, and I think Michelle's talk on mimetics in particular was interesting. Obviously, this is near and dear to my heart if you're familiar with the work that we published at Teneo Bio, using a similar approach, but using the heavy chain only transgenic animal to generate those VH binding domains. But I'd love to hear from Michelle just on some of the advantages that she sees of using that mimetic approach over maybe the engineered cytokines.
Yeah, so I think there's strengths and limitations of both. In terms of the strengths of an antibody-based mimetic, I think one of the main advantages would be we can utilize the binding diversity of the antibodies. So if we were able to create mimetics with very specific functions, then that could be one advantage over using a native cytokine that has a lot of pleiotropy. And then in terms of the limitations, there's not really, we can't really control the relationship, right, between the binding versus the function. It's kind of agnostic and needs to be empirically screened. And in terms of using natural cytokines, you don't have to be as worried about immunogenicity if you're just mutating like a couple amino acids to generate partial agonists, for example. One point I want to make is maybe one difference between the bispecific antibody approach that I use versus I believe you made diabodies, right, at Teneo Bio?
We use a bispecific antibody, but look more like a conventional antibody.
Yeah, so I think like the molecules that you were using would definitely have advantageous half-life, being, you know, much larger, and you even did, you know, in vivo studies and NHP studies. So
Yeah.
I think in terms of the antibody only approach, like that would be useful for preliminary screening, maybe, to discover like a whole diversity. But then like when zoning in on candidates, you definitely want to go with a full-sized antibody. Yeah.
I think those are all really, really good points.
Yeah, great. Thanks, Michelle. Okay, I think with that, we will conclude at least the formal session or formal portion of the cytokine webinar. First, I want to thank all of our speakers and panelists from today. Jeff, Nathan, Jamie, Andy, Michelle, thank you for your wonderful talks and taking the time out of the day to be here and present and also then meet with attendees after. And also a big thank you to Pam and Jan from the Antibody Society for helping organize the webinar, and also my co-host Kat. So I think, you know, we've learned a lot today about cytokines. I hope you've all learned a little bit more, and that they certainly are potent mediators of the immune system, but harnessing them therapeutically, particularly for cancer or even for autoimmunity, poses a lot of challenges. And we're starting to see this sort of resurrection of cytokine immunotherapy now with the more advancement in our understanding of the sort of basic biology of cytokines in the context of tumor immunity, which is exemplified by Nathan and uncovering a sort of a negative role of IL-6, and also novel engineering approaches to retain cytokines in the tumor, like Jeff described, or have cytokine activity on demand, as Jamie showed, and then also, you know, completely moving away to something that's a mimetic, that's not a engineered cytokine at all, and trying to recapitulate the activities or drive new biologies by sort of manipulating, using engineering approaches and non-natural ligands to drive new biology. How all these different approaches will play out clinically, I think will be very exciting to see over the next 5 years as we start to see more cytokines advance into later stage clinical trials. So with that, I think I will wrap up and just say a couple of housekeeping items. The first, for those of you that would like to meet the speaker, I believe Pam put something in the chat, but it will be a different Zoom link. So everyone should have received an email from Pam, and also the link to the new Zoom meeting for the breakout rooms for the Meet the Speakers is in the chat. So we will be disconnecting or shutting down this breakout session or this Zoom session, and joining another Zoom session for the Meet the Speakers. So again, thank you guys all for coming. Thanks for the speaking, and thank you all for attending and providing great discussion.
Thank you. Hope to see you all at the Meet the
Thank you.
Speakers.
Thanks.